thank you everybody for joining me and us on today. We're so thrilled to be here from Breakthrough Cancer, and this is the first official presentation that we've done with the team. So we're glad that you all made it to join us today. You all get like major brownie points. It's 4:30 on a Saturday and you're here, so thank you again. I want to give a special thanks to Kirk and the entire ALK Positive group, who we couldn't be more delighted to team up with. So I'm going to give just some brief introductory remarks, and then I'm going to hand it off to these rock stars because they're the main event. Yeah. Oh, that's true. So I'm Terry McGowan, the Director of Development at Breakthrough Cancer. And Breakthrough Cancer is a new foundation that was formed in 21 with an extraordinary gift of $250 million from the Goodwin family. And the Goodwins had been supporting cancer research centers across the country for decades through their foundation, the Commonwealth Foundation. And they brought everybody together, all the cancer centers together, and said, what could you do if you all worked together? So it was an extraordinary ask, and they said, you know what, we're gonna back this and support this. So they committed $250 million, and they gave the first $100 million free and clear to get us set up, but then there was a hitch. The next $150 million was set up as a match challenge grant. So the Goodwins, in their wisdom, knew that even though they were committing $250 million, that was not going to be enough to cure some of the deadliest cancers, that we were going to have to partner with other institutions, we were going to have to partner with other foundations, we were going to have to bring in other philanthropic dollars, and we were going to have to partner with industry and pharmaceutical companies and biotech companies. So 3 years later, we have invested $100 million into 4 areas: ovarian cancer, pancreatic cancer, AML, and glioblastoma. Our newest venture is the lung initiative, which you'll hear about. And basically, the mission of Breakthrough Cancer is pretty simple. It is based on the premise, what if researchers across the country who wanted to work together could actually work together? And it seems almost intuitive that why don't researchers just automatically work together? And the answer is actually quite complicated. Our medical research system is set up in a way that makes it very difficult for individual researchers at their own institutions to collaborate and share data on a regular basis. Not to say that there aren't wonderful investigators that are doing this on a regular basis, but it's difficult, it's challenging. So the Goodwin said, you know, we want Breakthrough Cancer to break down those barriers and work with researchers to make it seamless. So we decided that we would take a look at what the special ingredients for collaboration were. The first one was timing. So timing is everything. We lived through the pandemic, and the pandemic showed us that industry, academic institutions could work at breakneck speeds to bring new therapies and vaccines to patients. That was a clear indication that that was possible. We want to take the lessons learned from the pandemic and now apply them to cancer research. The next element is the right people. I couldn't be more proud to be sitting next to these investigators. But they're brave scientists who are thinking beyond their own institutions and their own labs and looking at the bigger questions. What are the real barriers that are preventing progress in a certain disease? And the last element is the environment. We want to drive radical collaboration and remove institutional boundaries. So that's a difficult challenge, but we're happy to say that we're on the way. So we developed 3 main goals for the foundation. One is to intercept cancer. I mean, this sounds so basic, but to intercept cancer as early as possible. And you'll hear from these researchers that minimal residual disease is an important aspect of intercepting cancer, specifically ALK positive cancer. Accelerate drug approval by learning in patients. So what does that mean? It means in early stage, first in human clinical trials. So instead of learning necessarily in mice or in a petri dish, to give the drug, get the samples, test the samples, and see in real time if the drug is actually having an effect on those cancers. And then also to develop new and interesting ways that we can work with industry. Academia and industry have not always worked compatibly and sometimes competitively. So we realized that as a foundation, we cannot bring new drugs to market. We need biotech and pharmaceutical companies to do that. So it's an important component of what we're doing. And I'm gonna try not to breach any confidentiality, but there is one large pharmaceutical company that we are talking to that hopefully will be working with us to develop a vaccine for ALK positive, focused specifically on MRD. So we got started with the idea of how are we gonna pull this off? So we started with the 5 institutions that we're currently working with. So Johns Hopkins, MD Anderson, Dana-Farber, MIT, and MSK. And we asked experts at those institutions, what would it really take to move the needle in some of these very deadly diseases? So what would it take? If money were no object, what would it take? And they all agreed that if we could remove the institutional boundaries and get people to really share data in real time and get virtual teams, which we call team labs, to really work together and share information in real time. And that's what we've done with our 8 teams that we've launched so far. We also asked the team labs to work urgently. So when we worked with the teams and helped them develop their individual proposals, we asked them, what are the current barriers and what are the hurdles that are standing in your way? And if money were no object, how would you address those? So that process was undertaken by the lung team, and they've done an amazing job. We have launched, as I mentioned, 8 teams, $100 million, and we're going to be investing more. We're actively fundraising for the lung team, and we're excited about the progress, and ALK positive. We couldn't think of a better partner than ALK positive. So I'm gonna stop talking. We're gonna save some time for questions and answers, so I'm happy to answer any foundation questions afterwards, but I'm gonna hand it over to you guys.
All right, I'll jump in and start and say it's been a real, it's really special to be able to work with Breakthrough Cancer, but let me start by saying what's the goal of this project, right? Because we've been talking a lot about research and we'll delve into the details of the project, but there's a simple goal. We want to turn ALK positive lung cancer into a curable disease. It's really that simple, important, beautiful, yet complicated. That's our goal. And you'll see how we're going to try to do that through use of really sophisticated technologies, through use of big clinical trials that involve ALK vaccines, and through use of some really fantastic partnerships across different institutions and across big pharmaceutical companies. But let me take a step back and say, first, why Breakthrough Cancer? And what is research? And I'm just gonna bear with me for 30 seconds, because we talk a lot about research, but not all research is the same. And why is this so important? A lot of the research that's done in university settings is funded by the federal government. And I would be remiss if I did not take this opportunity to say that, it is a time where where funding for cancer research is at an all-time low. So if we submit grants for cancer research funding, only about 1 in 10 get funded. And it takes about 2 years to get that funding. So 1 in 10 grants gets funded, and it takes about 2 years to get that money. That's really not a time frame that I'm comfortable with as a cancer doctor and as a cancer researcher to bring advances to my patients. So how do we make that go faster? How do we make advances come faster? And how do we really, understanding that it takes a team, it takes a village, no one person can do this alone, how do we make that happen? And part of the way is we've got to work together, and we have to find unique ways to fund research, because funding through the federal government will always, through the NIH and the National Cancer Institute, the NCI, will always be at the cornerstone of how we do research in medicine, but we need to think about novel strategies so we can make it go faster, so we can have more money for research, because this is how lorlatinib came to be. This is how all the things that we're talking about today are really coming to fruition through academic medical research. And hence comes Breakthrough Cancer. That is a big commitment of money with a promise for more over time and partnerships. Can't emphasize that enough, partnerships. So with that in mind, I also want to say, you're seeing 3 of us, but there's a tremendous group and like a really rock star, amazing group that's part of this from multiple institutions. I want to call out one in particular, who we all know and love so very, very much, Dr. Jessica Lin. Many of you know her. We all love her. We love her so very much. She could not make it here, and I know she's extremely disappointed, but she's been a very big part of this project as well, and I would be remiss if I didn't specifically acknowledge and say hi to all of you from her, because I know we've been texting with her today, and she's really disappointed to not be here. Let's go to the next slide. So maybe I will skip some of these and just cut to the chase, because we've all been sitting here for many, many hours, and it's time to go out and have fun in Nashville. Next slide. So let's start with this case, and I'm just going to go through the case, and then I'm going to hand over to my friends and colleagues, Vincent and Ross. So this is something that we're all familiar with, and we've been talking about it all day. We have good drugs, but they're not curing patients. We can talk about 5-year crown data, but we want 10-year crown data. We want 15-year crown data. We want more and more and more. We want to make this into, hey, you're living a normal life with your cancer. How do we get there? Is one drug going to be enough? Probably not. I don't think anybody in this room thinks that a single drug is going to be enough to cure cancer in the metastatic setting. So how do we take the offense against the cancer? How do we say, okay, I'm going to go on my drug and I'm going to have a good response to it, and then I want to do something now? At this time, we call minimal residual disease. So everyone who's on a TKI, a tyrosine kinase inhibitor, lorlatinib, alectinib, brigatinib, has gone on the therapy, hopefully seen a response, the tumor has shrunk, and then we kind of sit around and wait and say, let's wait for the tumor to progress. That's not good enough, and I think we can all agree that that's not good enough. So how do we take a position of strength and say, let's act on this when we know, why haven't all the cells responded to an ALK-directed drug? What's going on in those tumor cells that the tumor has shrunk? Maybe it's shrunk 50%, and we call that a success. Let's not call that a success anymore, because there's still 50% of the tumor still apparent on the CT scan. So this minimal residual disease state, which we've all experienced in clinic, where you've on a drug, you've had a response, there's still something visible on the CT scan, we don't know what that is. Why don't we know what that is? Because we don't biopsy the tumor at that point in time. And we just wait around for the tumor to progress, and then we switch therapy at the time of progression. So what we want to do as a team is say, let's not be complacent. Let's not wait until a tumor progresses. Let's not do the current paradigm, which is start a drug, see the tumor shrink, wait for it to grow. Let's take the approach of start a drug, watch the tumor shrink, intervene at the point of where the tumor has shrunk the most, at this minimal residual disease point, personalize a treatment at that point, and multiple approaches that we're thinking about, and actually take an offensive approach for the patient. I'm using a lot of sports analogies today. I'm not really sure why. I'm not really a huge sports fan, but anyway, it works. So this is our whole goal. And this is really a paradigm shift, because this is not how we practice lung cancer or cancer medicine in general right now. We tend to do one therapy, we watch it work, and then we watch it stop working. What we want to do is say, start a therapy, watch it work, understand how it's gotten to the maximal benefit point, but why isn't a tumor going from 1 centimeter to nothing? Why does it go from 1 centimeter to 0.2 centimeters? Why does it shrink 60%, 50%, but not 100%? How do we go how do we get to 100% tumor shrinkage so that we ablate the minimal residual disease state? That is really the crux of our goal here, and framing it in the context of ALK positive lung cancer. And I would venture to say that every single patient in this room has had this minimal residual disease state, where you've been on a drug, your tumor has responded, and then we just watch and wait for the tumor to progress. And we need to understand what's happening at that point so that we can actually intervene better at the time of minimal residual disease and get to the cure. I will hand over to Vincent.
I think Christina's done an amazing job just sort of outlining the approach and why we have picked the residual disease setting as the optimal setting to try to advance this this potential for improved survival and hopefully a cure. I want to take just a small step back just to also acknowledge why we are involved in the breakthrough cancer effort specifically. And to Terry's point about why this is really a novel consortium of investigators and a novel approach on how to do research and team science. In general, a lot of the issues that we are running into in terms of making advances in ALK rapidly really have a lot to do with obtaining tissue, obtaining blood at a large scale so that we can make the critical observations and generate meaningful hypotheses urgently and in a timely fashion. And you can only do that when you have a framework to collect and analyze these important patient-derived specimens that you all have played a critical part in donating and participating in these programs. We need to break down that barrier in order to make these advances at with the urgency that's needed. So breakthrough cancer allows us to do that because it brings together all of us and breaks down a lot of the barriers in terms of tissue exchange. There's a whole bunch of legal and other issues that are involved that get in the way of actually seamless transfer of tissue or data. And so this is really a vision of just trying to work together as if we were part of a single lab. And so that's what drew me to the collaboration, because while we are all deeply committed to trying to affect a cure for this disease, up until now, we've sort of been doing kind of our own things. And with collaborations here and there, this is an opportunity and a foundation that gives us the framework and ensures actually that we all row in the same direction. And so, we're really appreciative of the Breakthrough Cancer team for their funding of this, and also for ALK Positive for collaborating in this, because I think this is a worthwhile experiment to see if we can actually accelerate advances by working really more as a single team as opposed to separate teams. Ross, I don't know if you have any.
Is this working?
Yeah.
Yeah. Well, I'm going to channel the audience here and say, that's all great. What are you actually doing?
Who invited you? That's a great segue to this next slide. So the initial approach really is, and of course, we need to attack ALK at all different levels, for patients who have progressive disease, to try to find newer therapies, better therapies for ALK independent resistance, for example, where the TKI, lorlatinib or even the newest TKIs may not work. And so there are multiple efforts ongoing to try to elucidate some of those new directions. But for this particular project to start, we're focusing on this residual disease setting because we as a team feel that this is the setting in which we can most successfully affect a potential cure. And so part of the issue with this is that the residual disease setting is not well understood. And as Christine mentioned, in large part because we don't routinely biopsy your cancer once you've achieved a nice TKI response. So we don't know, the biology behind it in terms of potential drug-tolerant persisters and other sort of emerging mechanisms of resistance. So this is sort of a multifaceted approach to try to first characterize what residual disease in ALK positive lung cancer looks like. And so we have a tremendous preclinical team that's led by Aaron Hada at Mass General, Aaron Shank at Colorado, and others, Chris Love at MIT, and I'm forgetting others as well, but really to use preclinical models to better understand this residual disease state. And then we also have translational team to actually evaluate patient-derived specimens with the newest technologies, again, to better characterize what this residual disease entity looks like so that we can potentially propose therapeutic approaches to eradicate this residual disease more permanently. And then, of course, in terms of near-term deliverables to our patients, one of the flagship projects of this overall effort is to build a clinical trial framework in which we would enroll patients who are doing well with stable disease on their TKI. And this clinical trial framework would be at multiple breakthrough cancer sites. And this would give us a streamlined clinical trial that we can actually slot in various approaches to target residual disease. So vaccine being one of them, but there are others. There are different drugs being evaluated right now that may be particularly helpful to eradicate these drug-tolerant persisters. And so this is an effort to really just provide a framework where both patients can easily get on a trial to evaluate these potential strategies, but also industry partners and sponsors can actually have a framework in which they can seamlessly allow their molecule or their approach to be evaluated. Because, as Terry knows well, and even in our discussions with our own vaccine, you approach a lot of these industry sponsors and it's really a difficult proposition for them to actually do an ALK-specific study. And what we aim to provide with this project is to say, hey, we already have the study. We have the patients. And we have a systematic way in which we will collect tissue through biopsies. We have a systematic way in which we will collect blood to help, evaluate how well your particular approach works. And so this hopefully will accelerate new therapeutics, for our patients that are doing well in their TKI.
Yeah. Sorry, were you going to say something, Ross?
I was going to translate.
I was going to translate too.
Okay, you go first.
Okay, so we love you, Vincent. I'm going to use different words, that's all. So we've been talking a lot today about resistance. Well, how do I treat resistance, right? Well, how did we learn to treat resistance? We learned to treat resistance because we biopsied tumors when they became resistant. And we started to understand what resistance meant, right? So there was a time in everyone's life in this room where nobody got biopsies when their tumor became resistant. That was like earth-shattering, groundbreaking maybe 20 years ago. But through studies, it's like, hey, if we want to understand why tumors stopped responding to a drug, this acquired resistance setting, we got to biopsy the tumor and understand what the heck is going on. What changed? Why did the tumor stop responding? That was paradigm shifting, unbelievable. It changed the way we treat lung cancer, changed the way we treat ALK positive lung cancer in general. So let's put it in this context, because we want to do something transformative. We don't want some same old, you know. We want to do something that's really going to mean something. So again, I come back to this state that everyone in this room who's on drug has experienced. You go on a drug, the tumor shrinks, but it's not completely gone. Why not? If 100 tumor cells have ALK and the drug is against ALK, then why are 100 tumor cells not gone? Why is there residual tumor still left? Does anybody know the answer to that question? I don't. So if all the tumors have ALK and we give an ALK drug, why doesn't the drug work against all the tumors? Now, this is a really complicated question, and I'm oversimplifying it a little bit, but I would posit strongly that if we want to understand why we don't get 100% shrinkage, we got to biopsy the tumor at the time where the tumor has shrunk, but it's not completely gone. The time that we're calling the residual disease state. We do not do that as part of standard of care. If we want to be the offensive and say, okay, the drug worked and it worked pretty good, but it didn't go to 100% gone, how do we continue to make it go away? How do we treat and personalize at that state? We got to actually sample the tumor at that point in time, and we don't do that. So a big part of our study here is we're actually just going to sample the tumor at the time where the patient has had a response to their TKI, is doing well. We got to figure out, why hasn't the tumor completely gone away? What's happening at that time where the patient is stable on the drug, but the tumor is still visible? That's a big, big part of this study. It sounds so simple when I say it, but it's really actually very complicated, because getting biopsies at this time is not standard of care. Who's going to pay for it? Is it safe for patients? How do we get protocols in place for this? That's a big part of what this study is going to do, just getting the tissue. And then using a lot of sophisticated scientific stuff to actually look at, on a cell by cell by cell level, what is happening at this residual disease state. What do the tumor cells look like? What does the neighborhood of the tumor look like? So tumor is not just all tumor. There's other cells. There's immune cells. There's fibroblasts. Like, what's happening in the neighborhood of the tumor as well? And how is that actually modulating the response to the drug, so that we can say, in a strategic way, because we don't want to just throw spaghetti at the wall and see what sticks. We want to understand scientifically what's happening at this residual disease state. How do we intervene on it so we can go from a 60% tumor reduction to gone? Because our goal is to cure the tumor. Ross.
Oh, I can't follow that. That was awesome.
Back to Vincent then.
One of the challenges of actually doing a clinical trial in the residual disease state, and this has come up in some of these breakout sessions as well, you know, as part of the many insightful questions we've received, is how do you know whether or not it actually works, whatever intervention it is, whether it's vaccine or otherwise. And because many of these patients actually don't even have any evidence of disease on their scans. So part of the approach is really to develop ultra-sensitive methods to monitor disease in a setting where there's really not a lot of cancer, if any, active cancer. So, you know, that is one huge challenge of studying the space and actually conducting clinical trials in the space as well.
Should we open it up to some questions from the audience?
Maybe let's just go through the last innovation. Vincent, you want to talk about the
I
biospecimen?
want to talk about the infrastructure.
Yeah, before we open to questions, just one last portion, which is in addition to having these sites, because a big part of this project for us too is democratizing access to research, because not everyone can go to Hopkins or Colorado or Nashville or Houston or Boston. There will be certain aspects of the trial, like getting the research biopsies at the time of minimal residual disease, will be at the study sites, the major centers. But a big part of this too is wanting to really be able to, aggregate data and have participation across entire country and hopefully even beyond that. And so this part will be led by Jess predominantly, is to create an infrastructure for collecting data. And data is clinical data, biospecimens, blood particularly. And I'll just take this moment to give a huge shout out to Sophia and her team, who did such an amazing job today and yesterday in collecting blood. Wow, that's amazing. And thank you everyone who participated. That was an impressive show. But then also scans from patients, because another part of things that we're underutilizing is the information that we can actually get from scans, which y'all are having as patients anyway. And so collecting this information and using it and integrating it, clinical data, blood data, scan data, to be able to forecast responses is a big part of the study that we're about to launch.
So bringing it back to Ross's point earlier today about the good ALKs and the bad ALKs. So one important deliverable from this is really to be able to use this aggregated data collected at a national scale to make these key observations at scale in order to predict potentially who might have a good ALK or a bad ALK. And there are multiple questions in our breakout sessions about the use of AI. This would be one of them, for sure. And then I talked about this already in terms of the clinical trial framework that is being planned. And so, this is just an infrastructure that we're putting into place to allow efficient and accelerated evaluation of potentially promising treatments, with the vaccine being one of the first candidates that we're pursuing.
Vincent, do you want to expand a little bit on the vaccine? Because we had a lot of questions about your vaccine. You want to expand on this vaccine, maybe how it's different, what's the potential time frame for when it will roll out, just high level and as it continues to evolve?
Yeah, our vaccine study, which just got started recently in terms of treating our first patients, is a peptide vaccine. These vaccines that we're looking at are mRNA vaccines. Again, we had a discussion at some of the sessions about potential differences. It's not certain at all at this point whether one is better or worse than another. It's just different, and they engage the immune system in different ways. But we're really excited about the potential, and this is actually really a great example of how the Breakthrough Cancer Consortium and framework really accelerates our partnerships with industry, because as we approached each of these potential industry partners, the sort of the value proposition that we were able to offer really wasn't strategically compelling for them, except for when we got together as a group, as a consortium of ALK centers, and said, this is the framework we're building. And so that led to these potential partnerships you see here with Moderna, BioNTech, and others. And so we are planning on an mRNA vaccine for patients who are doing well on their TKI as the first clinical trial in this framework. It is still in the planning stages, so there's no set time frame for when this might be in the clinic and available for patients to enroll in. But we're hopeful that it'll occur sometime in the next year or so, as a very rough time frame. But it's very exciting in that it takes a slightly different approach than what our vaccine program does. But I think they're complementary approaches, and so it's really going to be very helpful to have these patient-derived data to help move the vaccine field forward. And again, I mentioned earlier, there's other non-vaccine clinical trials that we're developing as well for potential addition to this framework.
Yeah, and I'll just add to that. I mean, any trial under this breakthrough cancer perspective, because like Vincent said, it will start with an ALK vaccine, but we're building it so that we can kind of have modular units, like insert drugs as the science tells us, hey, here will be the best drug to treat at MRD with your ALK TKI, because like really the transformation here is we don't add a drug or a therapy at the time of residual disease, right? We kind of just watch and wait. You're on your ALK TKI. Let's wait for progression. We don't want to wait anymore. We want to give a drug, if you need it, to deepen and prolong the response to the therapy. Deepen the response, get a better response, get more shrinkage, and prolong the response. Make it longer, more years, more life. That's the goal here. The trial will be open, any trial here in this framework will be open in Boston, New York, Baltimore, Nashville, Houston, Denver. Those are the initial sites. So multiple options for patients is also our goal. We realize we'd love to have it at even more sites, but these will be the opening sites as we start to roll this out.
With the last bit of time we have left, it would be great, I think, if we open it up to questions. If you have any particular questions, we would be happy to address as best we can.
Hello? Hi. Sorry, it's not working. Sorry, Jane. You go ahead and ask the question.
It's working. When you're talking about getting to a point where things have been shrinking and then it stabilizes, and you're talking about a tumor that it shrinks, but then it stops shrinking over a period of time, is it the same for lymph nodes? Like, is a tumor? So when you're talking about tumor, it could also be lymph nodes. Would it ever make sense just to take those lymph nodes out?
You want to answer that, Ross?
Oh, Ross, we'll let you answer.
Well,
I'll run interference.
I mean, just imagine the scale of this. So they're going after a sub part of your disease, minimal residual disease, that nobody's ever studied before. So they've got to catch it, then they've got to analyze it, then they've got to find the targets, develop drugs, and then they've got to figure out how to measure whether the drugs work, because no one's ever done a study eradicating minimal residual disease. So if you're saying what the drug is, we don't know the answer yet. So to answer your question, yes, they'll take any tissue you can give because anything's better than nothing. This is transformative.
What is this they, Ross? It's we.
They?
They,
Them,
it's
them,
not
them.
they, it's we.
They, them. I've
We.
decided to use pronouns of they, them.
We, you are part of this. Okay, who has a question?
Hi.
I'll get you next.
One. No.
And then we'll take a question up front, okay? Okay.
Hi. I have two questions. The first one, if you had a progression, you treated it like with local radiation or you changed the therapy, and on the next scans, everything is stable. How long do you need until you can say this is a minimal residual disease from progression? This is the first question. The second question, in case you are in the minimal residual disease and you can get some tissue cells, how can you know for sure that these cells are persister cells and not resistant cells?
Vincent, you wanna?
Yeah, I mean, it's really an insightful question because that's actually part of what we're trying to learn. We don't actually know what the time frame is for, to Ross's point, we're all going to learn about this together. This has not been studied before. So when does residual disease occur? Is it when your CT scans start showing a plateau in your tumor shrinkage, or is it earlier? This is part of the effort to actually obtain tissue at different time points, blood at different time points, to really characterize this as best as possible. And the second part of your question was, how do we know if persister cells are
How do
persister
you
cells and not
differentiate
residual
between the
or
persister
resistance?
cell and the resistant cell?
No, it is a good question, because obviously, in our mind, and this is a slightly artificial construct, a persistent cell is, the hurdle, the turtle kind of, batten down and shelter in place. And then resistant cells are kind of stuff growing out from that. And there must be a blending which occurs, even before you see the growth on it. But again, without tissue, we can't even begin to answer it. So I think at the moment we throw the doors wide open. We capture, for example, the degree of shrinkage you've had, the time you've been on therapy, the time you've had stable disease, all of which might inform what we then see in the biology.
I have a question. If the MRD of a patient, aren't we all different? I mean, how do you do a vaccination for all of us? Do we have the same cancer cell structure, or are we different? Is that a stupid question?
Just to make sure I understand the question is, how can a vaccine work for everybody and not be specific or personalized? Yeah. Excellent question. I mean, we're still evaluating different vaccine approaches. So there's the concept of a personalized vaccine, where we actually grab some part of your tissue and then create a vaccine to target parts of your tumor that may be important to controlling your tumor. That's not what we're planning on doing in this first phase for this project. And then there's the concept of a shared vaccine, where it's a vaccine that is targeting something that should be available in everybody, all ALK patients' tumor. And so that enables sort of an off-the-shelf approach that doesn't require any sort of personalized matching. There's also a little bit of a nuance in terms of HLA matching, which I know some of you know about and have asked about. But that is also an aspect that may need to be sort of tailored.
Can you explain that, Vincent? What is HLA matching?
So human leukocyte antigen, it's sort of the fingerprint of your particular immune system, almost like a blood type, but really more of an your immune system type, which needs to match with particular treatments that you may get in terms of a vaccine, or what you may be more familiar with would be like a bone marrow transplant. So some vaccines or cell therapies need to have this HLA match. So we may actually have a vaccine that is HLA restricted at some point, but the initial proposal is actually a shared vaccine that would work for all patients.
All right, for sake of time, unfortunately, we can't take any more questions, but find any of us if you want to chat more about this project. It's about to launch. We're really excited about it. Thank you, Breakthrough Cancer, for your support. Thank you, everyone in your room, for their support, because literally this research does not happen without your support, so we're so grateful.